Welcome to the third lecture for regulatory science and translational research. How does one develop a project into the first in-human application and then forward? What animal models is one using and what challenges do we have here? The short overview of requirements, that will be a short part only, because then what are the principles? What is the mindset for non-clinical development? How does one plan this? And what do regulators think about this? I will share a little bit internal discussions with you because I think this is important to know also for you how this thinking is also evolving. And also, and this is actually the take-home message, what I would myself call a knowledge and risk-based brains-on approach. There's a lot of guidelines, you can see it here. This is not all of them. Design modifications of gene therapy, medicinal products, for example, a very important paper. Because the mindset was just in a nutshell that if there is a gene therapy under development and there is a change in this, in the vector, new promoter or a new antibiotic resistance gene, then this is a new product and then you have to start all over, which doesn't make sense, obviously. So there is actually a checklist in the legislation, and the legislation is here, this directive from 2009. There is a listing of what non-clinical requirements actually should be fulfilled. So for gene therapies, you can see here pharmacokinetics is not the standard pharmacokinetics, but rather biodistribution. But the rest is fairly standard. Genotoxicity, carcinogenicity, I will come back to this. A frequently asked question, do we have to do this? And what tests? The standard test is the Ames test, where you have bacteria and then you put the drug into this bacteria soup and then you see how many mutations are in there. I guess you all will see that this is not at all a relevant test. Nothing's going to happen. Unless there are some spontaneous mutations in the bacteria, so this is not a useful test. So this is already something where the normal testing procedures come to their limits. And for cell therapies, that's the same. Pharmacology, pharmacokinetics, toxicology, additional toxicity, very easy to put on paper, very difficult to do in practice sometimes, but of course there are also possibilities to do this. So that's the requirements, that's the starting point. But how to do this in practice? What about concrete products and how to do this then? And actually, what is very important is that one starts with at what stage the project is. Is it before the first in-man clinical trial, or is there already clinical trials ongoing? Or is it maybe a project which is already moving towards marketing authorization application? Because if it is an early project, then of course the non-clinical development has much more weight. If there's already clinical data, then it would sometimes not even make sense to come with another non-clinical study because there is human data superseding this. Some non-clinical experts do not like to hear this, but of course that's the truth. So we have to look at the stage of development and what kind of complementary data we already have. And very obviously, that requires a little bit of thinking because we have the two possibilities. We either test the product itself. That would be on the left-hand side. The advantage is that we actually test the product. All impurities from manufacturing and so on would be in there, and these also can be toxic. But the disadvantage is very obviously, as I said, that not all, if any, interactions are similar to humans. So in the worst case, nothing's happening in the animals, and then it would be the falsely negative conclusion on safety. That means that there is nothing happening, it's a safe drug. The other one is the homologous model. The advantage, as is very obvious, all interactions are relevant, very obviously pharmacology and toxicology, but we are not testing the medicinal product. So we would have to simulate the manufacturing process in this with the animal product. And that, of course, is a cumbersome procedure. And of course, it is sometimes a bit more difficult to do this. When do we then use this homologous model? Do we always use this? Do we always recommend this? Or is there a little bit more that we can say? Are there other approaches? And if so, like for example, newer animal models, would we accept them? And can we use existing knowledge from other products? Many groups have not only one project, but kind of a platform, maybe 2 or 3 projects. Maybe one is advanced already and they have data on this, and then they come with a second project. Do we then have to start all over again? Or can we use the data? And also from clinical studies, if there's already clinical studies. If there is a good reason to deviate from a guideline, it's still a guideline, not a law. then it is possible with a good justification. And of course, before one starts, one would have to kind of have an estimation, of course, what the relevance of the outcome is expected to be. We get scientific advices from companies, from also academic groups, and one could look into these scientific advices, which are confidential, very obviously, if there are common questions. Always the same questions, always the same answers. If that was the case, one could publish this. Then one would not necessarily have to come for scientific advice, one could just download this. Then these meetings in the Innovation Task Force, I've already mentioned this in another lecture, that one could use at the European Medicines Agency, but of course also the National Scientific Advice. And I'm planning to also give a lecture on very practically how this works, what you would expect if you would be interested to visit an agency like the Paul-Ehrlich Institute, for example, in Germany, or the Danish Medicines Agency if you want to come to Copenhagen, where scientific advice is for free, by the way. Then I could tell you how this is made and what you would expect and how this is done, how to prepare for this meeting, what we expect and what you can get as an outcome and so on. Normally the paradigm was every product is an own development. We look at this without anything else in isolation, but that is maybe not the best approach, especially in a field like this where it's so difficult to produce data. So that was actually, for me at least, quite an advancement, that also my regulatory colleagues, at least most of them, agreed to this. And then the developers industry was also there. They did not request the establishment of a different standard. So kind of, well, for HMPs, we cannot have the normal standard, we need something else, but rather to recognize the long-standing experience. That was especially the medical device industry, which is coming in with tissue engineering products, where they said, well, we have used this for 20 years. Now we are coming for a clinical development because we have to comply with the legislation. What about this? Do we still have to do non-clinical tests? And very obviously, the answer could be yes, because we have it in the guidelines and the legislation, or no, because we already have tens of thousands of patients being treated. So if the data was good enough from this, that would be, of course, fairly acceptable. Then what animal model to choose? That is maybe the most important question here. And it was very interesting to see in these discussions that there are kind of smart in vitro systems or ex vivo systems, like you put certain cell combinations together with growth factors into a medium and then have readouts on the cellular level. This is not an animal model, very obviously, but it is still testing in a very smart way the product. And that could actually, in certain cases, complement the non-clinical testing or even substitute animal testing. So that was quite good to hear this. And then we discussed on the homologous model, and this can be kind of agree where we use it, where we request it. And I think the common denominator was for a proof of concept. It is almost in all cases, or let's say usually, the homologous model. And this is also what you have in the publications. If an academic group makes a project, then they will have mouse data, for example, that this gene therapy or cell-based product or whatever works, and that's published. And that is the proof of concept study. So it wouldn't make sense to come with the human cells and then construct a very complex system that may or may not work. But of course, in an ideal world for toxicology, it would be best to test the product itself. Dose finding, that is something not that easy. I think what was said was in these meetings that the reduction of the number of doses to be tested could actually be possible, but may have to be compensated by clinical testing. But we normally request quite a range of doses, and that is sometimes, of course, not that easy. And for combined advanced therapies, that is cells, for example, together with a medical device, it may not at all be feasible to do dose finding, because the dose is something which is just one device filled with cells. Doesn't make sense to put 2 or 3 or 4 devices into an animal, but maybe here different fillings or something could be more informative. Risk-based approach that we already had, that is the most frequently used term, maybe also the most boring term because it's so frequently used, but I think it's the most important, maybe the most important milestone in the entire legislation, because it allows us to be flexible. Of course, the categorization of low risk and high risk, we had this already with the first-in-man clinical trials, should be avoided, because in the beginning, if we have a risk categorization of high risk, and it turns out to be a fairly harmless product, it still has the stamp of being a high risk. And who would invest into a high risk product? So that is something which we already some years ago left behind. But still, of course, we are talking eternally about high risk and low risk. And very importantly, the clinical context, at least in some people are very strong about this. The clinical context should also be an important factor to be considered. That means if after 1 hour of discussion, if this particular quality parameter is relevant or not, then somebody comes up and saying, well, if this works, this is for people with a deadly disease in the end stage of this disease, who cares actually if there is TSE risk or something, because the patient will anyway die unless we treat the patient. And that is something quite interesting also. It could allow also a certain way of argumentation around this, but at least it should not be forgotten. Many projects in gene therapy and also in cell therapy are for very severe diseases and not for common diseases. And this means that we may also have to take this into account when we talk about non-clinical studies, if they make sense or not. Products which also have been used in clinical trials and also in clinical practice. Some clinicians are using this. As you know, stem cells grow exponentially. Mesenchymal stem cells can be taken from the bodies, from the patient's body, and they can differentiate to different pathways, which obviously is the therapeutic intent, at least in some of the applications, but they also are immunomodulatory. And the problem that was discussed sometimes was if they could transform to malignant clones or not. There was some data published, but that was not reproducible, so it has not been clear if there is a risk with these products or not. So how to test this in an animal model? So the first, of course, would be what toxicology do we expect at all? There is behavior at the site of injection, so there could be kind of, at this point, if it fills for a defect, for example, and should differentiate into something, there could be de-differentiation or wrong differentiation, that there is cartilage in muscle, for example, just as an example. And what could happen at distant sites? Could it migrate? And here again, the autologous cells, of course, are much easier to migrate somewhere than the allogenic cells, because they would not be stopped so much. But of course, there is data also putting a little bit relative view on this as well, so it's not 100% true what I'm saying, but at least this is the easy thinking around this. And then we have the limitations of the non-clinical models, which are of course very obvious. As I said already, nothing might happen in animals. If you take human mesenchymal stem cells, some items or some readouts work, some don't, and the cells are immunogenic. So there could be an immune response against them, which would then kind of reject and kill them, and one would say, well, this is a very safe product, nothing happens. But of course, there are no cells anymore because they have been eliminated. And also there could be immunotoxic effects that would not happen in patients if the cells would act on the immune system. So there is this, by the way, this distinction, immunotoxicology or immunotoxicity and immunogenicity. Immunogenicity means that they are immunogenic, the immune system attacks them, and immunotoxic means that they are toxic on the immune system or interact with the immune system in a way that it is not intended. So here we have to distinguish. Another example, genetically modified T cells directed to attack tumor cells. That is very much also now with the CAR cells, the chimeric antigen T cell receptors, in the community. And here this is very obvious. Also the antigen recognition is highly species specific, as most of you, if not all of you, know. This is almost embarrassing to present this here. But if you take the human T cell receptor, of course this needs human co-stimulation and also the human MHC context to see the antigen, and the mouse MHC and the mouse molecules, of course, would not interact. And what are we expecting as regards toxicity? It is, of course, the directly expressed antigens in other tissues, but also this, what is called the degenerate recognition of the T cell receptor, the molecular mimicry. You know this from autoimmunity, that there is a virus infection, a viral infection or a bacterial infection, then the T cell receptor recognizes something which looks like it, but it's not the same, and this is a viral antigen, and then it kind of acts or reacts, cross-reacts against a structure of the own body. This has been shown, for example, for myelin basic protein, where you can see the sequence here on the right-hand side on the top in in the table, and here you can see some sequences of viral peptides which also activate T cell clones, although they are not human structures. So this is molecular mimicry, and the same could of course happen here. You have a T cell which is engineered to recognize a tumor antigen, but this could also recognize something else in the body. Testing in a homologous model could be, at least for proof of concept, be interesting. That's what is there. But again, for toxicology, that's maybe not informative either for the same reasons. Because a mouse T cell receptor would recognize something in mouse and not necessarily in man. So what to do? On this bullet point, CAT should explore the possibility of setting up a platform to share information with stakeholders on trends in non-clinical development of advanced therapies. And actually, two colleagues from the CAT and from its former working parties have made an analysis after 3 years of CAT, have looked at all scientific advices that were provided from the various committees on advanced therapies, and have tried to distill what the main messages is. And this is the link. This is open access, so you can read the PDF on the internet. Just in a nutshell, it was 54 advices overall. That's quite a lot. And 31 procedures contain non-clinical questions, so that is already a substantial number where one can look what the principles are. I think you cannot read this. You can see here, or cannot see here, that the main amount here was the green one, tissue engineered products and cell-based products, but also gene therapies, around a third of them, if I remember it correctly. And then also the gene therapies were also kind of subdivided adenovirus virus associated vectors, and lentiviral vectors were the main ones, but also some others. And also for the cell-based products, it was a bit 50-50. That's number C here, between autologous and allogenic cells. That maybe sounds a bit statistical, but it is interesting because it shows what problems actually the groups and the developers have when they design the non-clinical studies. You can see that the proof of concept was much clearer apparently for gene therapies. Only 36% had at all a question for advices. Okay, statistics, 36% is maybe not the best way of expressing this, but a third of them only asked, but 3/4 for cell-based products. So that was much more difficult apparently for cell-based products to do. Biodistribution was almost the same, general toxicity almost the same, tumorigenicity much more for cell-based products. That could actually be because either the gene therapies are not tumorigenic, because one knows it or there is at least some hints, or it's so clear that they are tumorigenic, or that they can have this insertional oncogenesis, that there is no need to ask for a question on this. But interestingly, a bit more than half of them asked if a reduced non-clinical package was possible. So apparently, this is a burning thing. So the trends, I would say in general, was that, and that is what I have already transported today, the message. It was recognized that conventional safety testing is not always feasible, or let's say only exceptionally feasible. A lack of a standalone toxicity testing is acceptable if there is sufficient data from the proof of concept studies. That's an important message, because that means if we plan this proof of concept study with safety built in, we can maybe come around another study that is actually saving time and money. And the lack of genotoxicity or conventional carcinogenicity is generally accepted. Like I said, these tests are not relevant. One doesn't have to do it. It's rather tumorigenicity, and then it's also more of a tailored approach. I'm coming back to this. Very practical, good laboratory practice, GLP, a big problem for many. That's the kind of, GMP for the production, but GLP is the same kind of standardized environment for non-clinical testing. And very obviously many labs are very high quality, but are not kind of accredited for GLP testing. If there is already a very nice publication on a proof of concept study which is not GLP, does one then have to repeat the study? Some people would say yes, because it's not GLP, but of course this is not always possible, and it would not make sense to repeat animal testing, because if it is published in a peer-reviewed journal, there should have been a lot of scrutiny behind this, and sometimes when one looks at the details, it is according to GLP principles, so it would be acceptable. So it is no problem in many times. Biodistribution, that's easy said, but difficult to be done sometimes. The duration of the studies must cover the persistence of the viral vector or include its clearance. But one can use intravenous administration very often as the worst case scenario for biodistribution. If it is a local administration into the muscle, for example, it can be very difficult to see biodistribution and where it actually goes. So this is why sometimes it was accepted to take intravenous administration as the worst case scenario, because then it spreads and then you can see that if there is a certain preference for certain tissues or not. And disease models and homologous models are generally encouraged for toxicity testing as well. So that opens the way to this kind of combined study that I've already mentioned. Let's very briefly, so that there's also time for some questions if you have it, briefly look at the cell-based medicinal products. Again, almost trivial. The choice of animal model was the most frequently discussed item. In general, preference for a homologous model mimicking the clinical setting. The cell-based products are, for example, tissue-engineered products, which would be locally applied to the heart or somewhere else. And this should mimic the clinical approach, and that can be quite difficult, because to apply cells in a living mouse to the heart is not that easy. And if it is a combined advanced therapy with a medical device, it's maybe not possible at all. So then, of course, this can, but not necessarily has to include a large animal model. One would also would encourage the use of more recent in vitro approaches. There is a lot of research going on in this, and this could complement or even replace testing very elegantly. And here we are back with the topic that how regulatory science and basic science also interact, because basic science will create these approaches, and then we have to learn what that means, and we have to kind of accept them and implement them in our system. So these product-specific risk factors, this is the way forward to look into this risk-based approach and towards a more tailored approach to this. So that's what I meant with the knowledge and risk-based brains-on approach instead of a tick box approach. Where do we have to do more? Where do we have to do less? That's the central question for the risk-based approach. Use the non-clinical models more in a directed approach, and what is it that you wish to show and how do we best do this, and make use of the regulatory authority. Because we have seen a lot of products where we have maybe already encountered the problems and solved the problems with other groups and other people that this particular group now has for their project, and that maybe saves a lot of time and resources. This is a little bit of a summary of this. We have the non-clinical development proof of concept, and here I have just put on the left-hand side the quintessence of what I have just talked about, what model to use, and toxicology. Check before you start, and check if one can at all test this, and check with knowledge from the other studies and also from literature, and then combine this with smart in vitro and ex vivo testing and disease models, together with a risk-based approach, of course, and the stage-based approach. If there is already a lot of data from other sources, one may be able to skip this. And I think here I close, and thank you for your attention. If you have any questions, please feel free to ask them.